Okay, so welcome everybody. This is the TIA Centre seminar series. Today we're joined by Mattias Rantalainen, who's going to be giving a talk on his recent work. So we're very lucky to be joined by Mattias. So since 2013, Mattias leads the predictive medicine group at the Department of Medical Epidemiology and Biostatistics at Karolinska Institute in Sweden. He completed his PhD at Imperial College London and also did a postdoc at the Department of Statistics at the Wellcome Trust for Human Genetics at the University of Oxford. And more recently, he's now involved very actively in the area of computational pathology. So we're very much looking forward to your presentation today. And whenever you're ready, please share your screen. Is it okay if people ask you questions during the seminar?
Absolutely. Feel free to interrupt me. Is it correct that the presentation is 35, 40 minutes or something to leave plenty of time as well? So I think in that case, definitely just interrupt me and ask a question if you feel like them, like it during the presentation.
Sounds great. So for those online, if you want to either raise your hand or type in any questions in the chat, both are very welcome. So with that, you can share your screen when you're ready and go ahead.
Sounds great. Thank you so much. Thanks for the introduction and thanks for having me on here. So let's see if I can manage to, hopefully you can see a full screen rendition of the slides.
Yeah, it looks great.
Okay, perfect. So great, yeah. So today I'm going to talk about some of our activities in the area of computational pathology here at KI. So my talk will have, I guess, two parts. The first one is to give a little bit of an introduction to how we think about this domain, and then the second part is about more specific research studies and activities that we had over the last year or so here. But I assume since this is a relatively, I guess, specialized forum and seminar series, so I'll pace through the introduction quite rapidly, but just to give you some kind of firm ground to stand on, still have an introduction. Okay, so let's see if I can manage. So these are my disclosures. And before I start, I just want to acknowledge everyone in the team as well, who's actually done the research that I'm going to talk about today, particularly Yinzi Wang, who is lead authors on two of the studies. So the background to our work is, I think you're mainly familiar with, most of you, the two trends that have happened over the last years or maybe decade if you're you look a bit further back as well. But one of the trends of the transition from analog traditional kind of pathology workflows in the clinical setting to digital workflows. So that's happening quite rapidly in many places, but particularly in the Nordic countries, and I think in the UK as well. So I think at least in Sweden, all 30 odd pathology labs are digital today, and they don't necessarily work fully digital, but they have a digital capacity and scan basically all slides today, I think. And in parallel with this development, there's also been a rapid evolution of deep learning methodologies that has been particularly effective for computer vision image analysis tasks, and that's been emerging over the last, I guess, decade almost now, but since 2012, 2014 and onwards. So what we see is obviously the potential, as many others see as well, to work with different type of deep learning solutions or image analysis solutions to provide decision support to pathologists in their kind of routine work. So we don't we're not as interested in this fully independent kind of model-based approach, but we rather see that there's the greatest potential is to have this as working as a tool to help the pathologist in their routine work. So at the bottom right, we also I tend to make a distinction between two areas of computational pathology. The first one is problems or kind of activities that support routine pathology applications. And a lot of progress has been made in this domain for detection of cancer. There have been numerous studies published in this, basically where the performance is on par or even better than humans are today. And there are, of course, a lot of reasons to engage in this, and we'll come back to that a little bit later on. But it's essentially doing the same thing as the humans are doing today, but with higher efficiency and slightly improved quality. But the second point in the bottom here is what I think is most exciting about this space, and that is precision diagnostic solutions. So I define this just as approaches where we go beyond what can be done today in the routine pathology setting by human specialists. So essentially extracting information that is beyond the routine, and that could be prognostic information or predictive information, or predicting something else like biomarkers, so from the images, from routine stained images directly. Okay, so the other background slide here is just to clarify the precision diagnostic part a bit further as well. That's how this fits into precision medicine. So there's probably many of you also well familiar with this, but conventional medicine has, in many cases at least, seen patients with particular diagnosis as a kind of homogeneous group, and there's been a one-size-fits-all kind of strategy to treatment. But obviously, particularly in cancer medicine today, there's a precision medicine paradigm that's been around now for many years actually, but where we actually acknowledge that patients is a heterogeneous group, even if they have the same kind of same diagnosis on the high level, breast or prostate cancer, whatever it might be. But on a molecular level, these patients are still different, and that's revealed both by molecular profiling as well as the probability of responding to particular treatments conditional upon certain point mutations or other characteristics. But the key part here is to enable precision medicine, we need to do some phenotyping. And the main mode today in precision medicine is molecular profiling. But the less explored domain is to use routine histopathology images here as well to extract phenotypes, measure something, some representation, something that we can actually use for stratification of the patients, and ultimately in the future, perhaps also help guiding treatment decisions. I don't think I have to talk to, this is just about defining terminology. Digital pathology we see as the kind of infrastructure part, just getting the lab to work on a digital workflow, while computational pathologist activities where various type of modeling and computer vision techniques are applied to understand or analyze the images. And in this seminar series, I'm sure you already covered some of these papers, but there are numerous others, of course. But it's actually in quite recent time, although computational pathology or computer vision applied in the pathology space been around since the 80s, maybe even earlier. It's just over the last couple of years, I think, that's been really impressive studies. Obviously, Nature Medicine here by Fuchs and his team in 2019, what was a clearly seminal paper. I think the two papers here on the bottom as well that highlight this ability to extract some molecular characteristics directly from H&E images are also very exciting to see, and kind of highlight this opportunity to go beyond the human eye and human mind. Okay, so now that was the kind of general background, and then I have a couple of slides about what we are interested in and what we're working on at the moment, and then we'll go into the actual science and our results. So we work essentially across these three different domains. Routine pathology, we do some of that, but it's not our primary focus, sorry. The second part is the AI precision diagnostics is perhaps our main focus and main interest. And then on the third part here is about providing standardized ways to extract phenotypic information from images. And we think this is probably going to be important in the future to enable enable people and studies and trials to measure things in a consistent way. So you can at least within a certain diagnosis measure the same thing across multiple contexts and compare the results and use information almost in a similar way as you would with molecular phenotyping data or clinical phenotypic data. So here at Karolinska Institute, I've led this effort called CHIME, the Cancer Histopathology Image Epidemiology Project. Bite size of a title here, but what we do in CHIME is to establish large retrospective cohort studies where we have we do the study design. We do everything from scratch, basically. Study design, we collect data, we access archived slides. So the retrospective part is basically that we're interested to having patient like clinical follow-up information. So we go back in time, like a decade or so, extract archived slides, we digitize them, and then we also link them with the registry-based health data here from various type of registries, but there are quality registries in Sweden which capture both outcomes and some other clinical information that can be directly used. In some studies, we also have molecular phenotyping data on the same patient, so everything, all the different data modalities can be matched up. So we have this kind of data collection, study design part of our activities. But then the main work in the research group is, of course, to apply and develop different type of machine learning and AI, deep learning type approaches to extract information and do studies in this domain in areas that we think are exciting and interesting. So today we have a capacity to digitize about 5,000 slides per week, and we're kind of gradually scaling up our capacity as well to collect more data. And we think that data is really important in this space. I think someone raised their hand. So here in pop. So
yeah, that's just me. Just a question. So it's a really fascinating amount of data that you're bringing into your center. So what kind of rate are you also able to collect the associated clinical information?
Sorry, I didn't hear you. At what rate or?
Yeah, exactly. So of course, you're getting 5,000 slides per week, you said. If you want to train your associated deep learning models or whatever kind of models you have, you need to have that associated clinical information, don't you? So I'm just wondering at what kind of rate you're also collecting your label data.
Right. Yeah. So we will come back to the exact labels, but on a patient level, such as treatment information, clinical outcome information, etc., we're able to kind of link these data, not week by week, but we do it in a kind of a batch way. But we can do that continuously. We do updates of the data sets maybe every quarter or so. So we tend to collect data, link it to the registry information, clinical information, and then after that we have different versions of the studies that we can pursue research with. So yeah, so the main, 5,000 slides doesn't mean 5,000 patients, as there are also multiple slides in some studies per patient, but we tend to have clinical information accessible.
Okay, perfect.
For most of them. Yeah. Great, thanks. So at the moment today, I'm going to talk about results mainly in the area of breast cancers, but we are also running studies in other solid cancers, but it's mainly kind of cancer oriented, our activities to date at least. And we have also close collaborations with clinicians, and we also do multi-site studies to some extent here in Sweden. Okay, yeah, so this is the kind of also a bit about the context here. So a lot of the research, of course, kind of early discovery, conceptual research. So development and validation of solutions in the space and demonstrating what can be done. But we will also have interest towards the translational side, where we particularly through a project called SWIPE, Swedish Air Precision Pathology, which is it's funded by the Swedish Innovation Agency, a governmental body, where we have both industry partners, several, like 3 healthcare regions today involved, where we actually implement and deploy some of these solutions, both for research and for clinical use. So we have this span from early stage research all the way through translation and implementation as well.
I'm not sure if you've seen a question online, Mattias, but
probably not. Can you read it?
Yeah, of course. How do you select the H&E slides per patient? Do you scan all slides or only selected positive slides?
Yeah, great question as well. It depends a little bit on the study. So we have multiple studies. Sometimes we only scan So in some cases there is an indicated primary diagnostic slide that we can easily find out. And in those cases, we from some of the sites, we have decided just to scan that slide plus other IHC slides, for instance. But in other cases, it's not possible to know what the primary slide is. So then in those cases, we actually digitize all slides from the patient, which can be quite a few, so between 10 and 30 for a breast cancer case.
Okay, great, thank you.
Thanks. So moving on, so the methodological part here, I'm just going to talk super briefly about, because I think you're all, most of you at least, are familiar with this. We tend to use fairly standard model architectures. Sometimes we do a little bit of changes to them, but we are not in our primary game is not to develop new architectures and so on. It's more about, I guess, applied research, but then sometimes we also go beyond that and do some methodological development if we think there's a need for it. Most of the work is kind of supervised or based on self-supervised kind of modeling, contrastive learning, and so on. There are weakly supervised learning paradigm means just that we have patient level labels, but we don't have pixel level labels. So that depends on the kind of objective of the studies. But in many cases, there are only, by definition, there's only patient level information available, such as, you know, predicting prognostic information. The patient can only have one global outcome. Of course, we couldn't have pixel level outcome of that. So that's important to know. The modeling, I guess, for those of you who are not working with deep learning, if there are any one of you who are not familiar, it's basically very large, complex models that can learn representations from in a kind of automated way almost. And the objective function for this is conventional, different kind of loss functions that are used in statistical modeling more broadly as well. But with the caveat here is, of course, that you need a lot of data to train these like highly parameterized models, and sufficient compute resources as well for that. But I'm happy to discuss particular questions if you have later on or now, but that was like the brief methodological statement, I guess. But it looks like this. We have a kind of a semi-standardized pipeline for pre-processing the images, which is not anything surprising. It's tissue segmentation. We tile the data to small image tiles or patches. We tend to do color normalization, and maybe there are discussion points around that as well, but we think that's can be helpful. For most of our cases, then we have a tumor detection model. So instead of having a human annotating the part of the slides that include invasive cancer, we have a trained model that do that for us to allow us to scale. And eventually we have a set of tiles basically that come out from the invasive cancer part of the slide. And then we fit various type of models or do different type of research analytical activities after that. So this is just to illustrate what it looks like. The work I'm going to talk to you about today is from surgically resected tumors from breast cancer. So they tend to be quite large in areas, but they're not as small as core needle biopsies at least. So we have this tiling strategy. We have a model to detect the invasive cancer part, and those are this is the area that we then continue to do further analysis after that. And there's a raised hand as well.
Hi, Mattias. Just another question that's come in from Nasir, and he asked, do you have to do tumor segmentation for every downstream task?
Probably not. We know that it depends on the task, but in many cases it makes sense from I guess from a medical and biological perspective to focus on the tumor area. But for some models, for instance, for risk, like prognostic models, for instance, could probably work without doing that. So I think it's a bit context dependent.
Sure, thank you.
So this is, I think, is an interesting discussion point as well. What is the current kind of primary limiting factor in the field for getting, high performing models in this space? It could be, I mean, there are of course more axes than two, I guess, but the primary ones are a methodology, like how are we doing? Do we have sufficiently good strategies and methodologies to model the data? And on the other axis, we have the amount and quality of the training data to optimize these models against. And of course, I think we're probably limited in both directions. I think looking at many publications today and also our own work, we think we're primarily kind of limited by the amount and quality of the training data still. And exactly where it's kind of diminishing returns, of course, with more data, but it's clear that many cancers, at least, are heterogeneous in several different aspects. So there's kind of a long tail of rare phenotypes, and given that, there's clearly a need to have quite large sample sizes to actually get the best possible model. So we think that data matters a lot actually in this space still. So now I'll try to go through two of the studies that we have recently worked on. The first one is focused on risk stratification of intermediate risk breast cancer cases. So in routine pathology, there's the Nottingham histological grade system, which grades tumors in 3 categories: grade 1, 2, and 3. About half of the patients end up in the intermediate risk group, which is the grade 2 class. And among those patients then that are ER positive and HER2 negative breast cancer patients, so these are biomarkers that relate to therapy decisions as well, which is a fairly large group. They have a relatively inconclusive diagnosis, and there are no clear indication necessarily for the treating physician on how they should be treated with respect to adjuvant chemotherapy. So our hypothesis here was that we wanted to explore if we could risk stratify this intermediate risk group patient further by using image analysis. And we took a fairly straightforward and simple approach to this. We trained a model that to detect morphological patterns in the grade 1 and grade 3 groups. So we basically hope that we can learn representations that relate to grade 1 and 3, which are low and high risk groups. And then we subsequently apply this to risk stratify the intermediate risk group patients into 2 different risk groups, one with high risk and one with low risk for progression in the disease. And this is a study based on around 2,800 patients in total, of which 1,200 belong to an independent validation cohort. So the results are up on the screen here. So on the left, you have a couple of Maya curves from the NHG kind of clinical routine stratification, where the green line in between here is the group of patients that were grade 2 and that we want to risk stratify. So on the right, you see a couple of Maya curves again for just those patients in the green group on the left. So those are the grade 2 cases. And it's clear that we can, with the help of this type of modeling, can group them into a low and a high risk group. And we also then continued to do a multivariable survival analysis using a Cox proportional hazards model, where we also adjust for other risk factors. And we have a estimated hazard ratio here close to 3 and a significant p-value as well. So this looks pretty good in the, now I don't have the validation set, but we also confirmed this in the test set, and the hazard ratio was slightly lower, around 2, but still significant in the external validation cohort from a different site. We also inspected in the study whether routine markers for proliferation, such as Ki67, were different between the deep grade 2 low and deep grade 2 high groups here to see on the left top panel, and the Ki67 were not different here in terms of that's marker. And the top right panel show the intrinsic subtype distribution here, and the interesting part here is that the grade 1, the clinical energy grade 1 group share very high similarity with the DG2 low group, which is the low risk group from our model with respect to the breast cancer intrinsic subtypes here. So there's a lot of similarities between this. So we think that the model is actually doing something sensible here. Yeah, and there's a raised hand.
Yeah, just a question. If you're getting this nice stratification, or it seems that there's this correlation with Ki67, what would you say? Would you say that you could equally just use Ki67 to perform the same stratification?
Exactly. I mean, it is exactly that we wanted to explore here, right? To confirm that Ki67 was not redundant, provided exactly the same information as the model, but that doesn't seem to be the case. No, exactly.
But I guess you wouldn't have to use Ki67 then if you can perform that.
Oh, right. I see. No, I think it's we would certainly not say that at this point in time. Rather the opposite, right? So you can't argue that Ki67 would be able to provide the same information as the model we have.
Yeah. Okay. Perfect.
Yeah. So this was a very short summary of this study, but we think this is a very promising example of where we have precision pathology. We could extract information, in this case, prognostic information that is not possible by performing routine pathology assessment here. And we have additional validation studies ongoing as well here to build additional evidence. And there's a regulatory approved product that will be released just in a couple of weeks now as well for this solution with a CE-IVD approval. And the second study that I hope to walk you through was published in Cancer Research last year. And this is less clinically focused, but more focused on kind of a conceptual idea of being able to predict molecular markers here in the form of mRNA expression directly from H&E stained histopathology images. So what maybe we can say a couple of words. There've been a few studies like this published so far. We took a slightly different approach to the previously published ones, in the sense that we don't rely heavily on transfer learning. There've been various approaches, but it's of course very computationally demanding to fit individual models to each gene in the transcriptome. There are around 19,000-20,000 genes. And what we did was actually to assess whether individual models, we basically optimized individual models for each gene in the transcriptome. And this is also weakly supervised, in the sense that we have one global gene expression profile for each whole slide image. And then for each gene, we fit one regression type CNN model. And it required a lot of compute hours, 300K GPU hours roughly. We found that about half of the genes had half of the genes had a prediction from the CNN model that were significantly correlated with the actual RNA-seq expression, also after adjusting for multiple testing by an FDR-based approach. So that's interesting in itself, but many of these genes, of course, those associations can be quite weak, so they don't have necessarily that large effect sizes. But we brought forward around 1,000 genes based on the effect size, Spearman rank correlations, validation in independent data, and about close to 90% of these were validated both in internal and external test sets. So we think that's quite encouraging. Of course, we don't think that this is going to replace RNA sequencing anytime soon, but it's an opportunity to actually extract predictions of gene expression for a substantial number of genes. Of course, the underlying hypothesis or assumption here is that there has to be a morphological change, right, that is associated with the gene expression abundance, and that's obviously not true for all molecular markers or for all genes, but certainly in some cases it seems to be the case. So this on the right here, you can see histograms of the Spearman row correlation coefficients in the two test sets. So you can see that the median correlation between predicted and actual observed expression is around 0.4 or so. So that's depends on how you see. It's like it's not close to zero, but it's not of course perfect neither. So it depends on the application whether this is useful. And the next thing we were interested in was to see if also we could predict spatial intratumor spatial variability, and that is relevant because a lot of like intratumor heterogeneity is driven by various kind of molecular mechanisms and during the cancer evolution, and it's also associated with increased high heterogeneity, associated with increased risk for developing drug resistance or having a worse outcome. So it's something that it's worthwhile to see if we can actually measure and quantify. So due to the fact that the CNN modeling approach is weakly supervised based on tiles, we also get, in fact, tile level predictions of expression. So we can create this kind of spatial organized representation of predicted gene expression that you see on the top right, directly from the H&E stained image. And to evaluate whether this actually worked, we performed wet lab-based spatial transcriptomics using nanostrings geomics DSP platform. And then we can get, this is based on regions of interest. So we in this case, we profiled 12 ROIs per slide in this study for the validation. And then you can look at how well they correspond, the predictions in those ROIs versus the actual spatial transcriptomic measurements. And at this point in time, when we did this study, there was only it was kind of a pre-release of the non-string platform. So we only had access to a panel of 76 genes that are oncoimmune and oncoimmune panels. So it's kind of relevant genes for cancer. We have 22 slides, 12 ROIs per slide. And we looked at the association, again, spatially then between the predicted and observed expression using a linear mixed effects model with a random effect that captured the between slide variability. And in this case, 77% of these genes also were significantly associated then in a spatial sense. So I think that's also pretty interesting. We did another orthogonal validation of this, just looking at immunohistochemistry. And in this case, we had the Ki67 marker, which is a proliferation marker that we mentioned previously. So that's like routine pathology. And then we predicted a proliferation score. So this is the proliferation score is based on gene expression in a set of genes. And we look at the predicted gene expression score. And the expectation here is that there's a correlation between the predicted proliferation score and the Ki67, which is a single proliferation marker then, but it's measured by immunohistochemistry. So we have these kind of rows that are paired, and they are grouped also by different subtypes. So you have to look at them row-wise and then judge for yourself if you think there's any spatial concordance or not. But at least for the proliferation score, there's a correlation, a quite substantial correlation between the predicted and the actual proliferation score defined by RNA sequencing. So again, we think this is looking fairly promising for some applications at least.
Mattias.
Sure.
Great. So we have one question online. I think the previous slide did definitely dig into what the question was alluding to, but we have a question that asks, in your gene expression work, have you done analysis on what kind of patterns are correlated with the expression profile of a given gene? So for example, on the previous slide, you have some of the correlation coefficients are very high. So have you potentially discussed this with the pathologists and tried to identify which morphological patterns have been the reason for this high correlation?
Yeah, I think that's an excellent question. And we have done a little bit of work on that domain, but I think in many cases, there are, of course, there's some easy ones where we have gene expression of genes that are associated with the immune response, for instance. And if you have immune cells enriched in that local area, that's a connection that you can make for sure and have a plausible hypothesis about. But there are also a lot of other genes that are not as directly related to a particular function or cell type. And the tricky part here, I think that in many cases seems to be very subtle differences. So looking at by eye, it seems very, very hard to come up with these kind of clear explanations about something. We also attempted a little bit of different methods for explainability or interpretability of these deep learning models. But the sad news, I think, both in this context and in many other contexts, is that the different methods that are available and fairly mainstream are not very concordant. So we think that at the moment, the methods and approaches for interpretability on a local pixel level scale is not really reliable enough to make conclusions in that ways. But of course, it would be highly desirable to try to come up with these kind of human understandable morphological patterns. But at least we are not there yet. But maybe others are more successful than we have been.
Yeah, I wonder whether you could potentially extract some handcrafted features within those highly attended areas and try and analyze those.
Yeah, that's a good point. There's one PhD student in the group, Philip Weitz, who are currently looking at that, not in the context of gene expression, but in another study. So that might be a viable way to look at cell shape and size and other type of traditional morphometric measurements. Yeah, it's a good suggestion.
Right.
All right. So I think we have. The last bit I wanted to show you. I think we already talked about this. So the last thing, we're now having another follow-up study that is based on a 5 times larger data set as well, with around 6,000 patients. So we think that will be add even further information about the reliability, and maybe even additional genes can be predicted with more training data as well. But a follow-on study on this kind of intratumor heterogeneity work here is also being pursued, and that's mixing these kind of looking at, we predict gene expression by the models that we produced in the previous study I talked about here, and then so we look in a set of genes that are included in a gene panel called PAM50, that is a breast cancer specific gene panel. We predict the gene expression for all those genes spatially. And then we apply traditional computer vision methods for extracting various texture features. So we kind of collect all these kind of texture features for each gene. So 50 times the number of texture measures, plus the gene expression as well in itself. And then we apply time to event analysis, survival analysis again. And the hypothesis here is that basically we think that it's possible to capture and measure intratumor heterogeneity for these 50 genes in a way that might be prognostic. So that's the hypothesis we wanted to explore here. And in this instance, we dichotomized the risk score that came out of the survival model in 2 groups. And you can see the survival curves here in the Kaplan-Meier format to the left. And we also performed a multivariable analysis here. And the point estimate of the hazard ratio here is 2, which is also significant by means of the p-value. So on some level here, we first predict gene expression spatially. We then summarize the spatial variability using traditional texture metrics, and then develop a model that is prognostic based on that. So that seems to indicate that there's some level of prognostic information also in this kind of spatial information in the tumors that is in addition to and provide prognostic information beyond the routine markers here. And this is just some visualization about this, but I think we can perhaps skip that. And the absolutely final slide before I stop is another example in prostate cancer, actually this time, where we have a precision diagnostic solution, where the purpose has been to see if we can increase sensitivity in the biopsy procedure for prostate cancers. And the hypothesis here is that there's at least some fraction of patients where the systematic biopsies are missing the actual tumor lesions. So on the left image here, you see the red areas, which are supposed to symbolize areas of invasive cancer. But then the pattern of the biopsies might, in fact, miss them. And then the question is, if you have a biopsy that might be perhaps nearby an actual cancer area, but doesn't hit it. So formally, when you look at this, looks like benign tumor tissue, but we hypothesize that there might be subtle changes still in the tissue that is nearby, that through a kind of computational modeling approach, we could maybe extract. And you see the results here, the ROC curve. It's not immensely impressive, but it's still an AUC of 0.7, 0.73 actually on patient level to detect cases where there's a risk of cancer despite having benign biopsies. And this study was based around 15,000 benign biopsies. But I won't go into the study design further here now unless there are any particular questions about that. So my general concluding remarks are, I think it's a super exciting field, and I know a lot of things are happening on your side as well in this domain. But the kind of main core here is that I think that there are many things where we could apply these techniques, everything from routine pathology tasks, but also to do more precision diagnostic studies and develop solutions in that space. We need a lot of data and well-designed studies to move this field forward. We work on some studies ourselves, but we're also happy to collaborate with you and others to build evidence in the space, basically, and do that together. I think that was everything from me, and I hope obviously we have maybe a few more minutes for questions if there are any.
All right, thank you so much for the fantastic presentation. Yes, you are right, there are some questions. There's a few in the chat. So if you're happy to answer those, that would be great.
Yeah, okay, so let's see here.
So I can read them out if you would like. So there's one question that goes back to, if you remember, we were discussing about the interpretability and explainability of your results when you're predicting these different mRNA expressions. So there was one question that basically he was asking whether there was any correlation potentially with different types of inflammatory cells. So he was asking whether or not you've got access to some different inflammatory cell annotations and whether you've thought about trying to map the expression level to what's going on in the inflammatory cells.
Yes, that's a good question. So we have not got access to annotations at the moment, but we have looked at quantification of TILs, for instance, a little bit, and do that in an automated way and looked at correlations. So there's some kind of superficial work that's been kind of initiated, but we haven't done it in a comprehensive, broad way to answer the exact question that you are suggesting. I think it's likely that there probably will be some associations, and that will, of course, approach the interpretability for some subset of genes as well. So I think that's a good suggestion and comment.
Great. Then the next one from Nasir, and he asked whether you've looked at the relationship between color-based features and gene expression, and pointed a paper that we've published in the group where we've essentially just used the color information to predict the gene expression level.
No, we haven't. So that's also in the H&E images. No, that sounds exciting. Right, color features. No, I will definitely look into that. We haven't looked into it, but that sounds exciting. Or maybe that seems like a potential problem then, if you can just look at the color profile. I don't know.
Yeah, I guess for specific tasks such as, I believe, HPV prediction in head and neck cases, the color is really important. So I guess whether that translates to other tasks as well, such as predicting genetic expression.
Yeah. So what we have done is to verify that not all of these 1,000 genes are highly correlated with each other.
Mm-hmm.
So there is like multiple, there's some like kind of latent structure. They're independent components of expression variability here. So I guess if the color was the driving factor, that would be just a one dimensional or low dimensional information, I guess.
Really. Okay, so Dang Online asks, which magnification level and size of patch did you use when training your models?
Yeah, exactly. That's also a very good question. We have used 20x resolution. We have optimized that a bit to seen it as a hyperparameter to evaluate before we done the studies. But particularly in the gene expression work, there's obviously a really high number of models. So I can't remember exactly how many genes we looked at when we picked one resolution level, but I think that it's likely that either a multi-resolution model could work better, where you take input from multiple scales in the same model, or alternatively that you actually optimize resolution for each individual regression task. So it might well be that some genes could be predicted better at a lower resolution, where you capture more structural aspects of the tissue, while others might benefit from a high resolution if it's more about single cell morphology or something like this. But in our case, I think that the patch size is about 270 micrometers or something at 20x. So it's like, I think 598 pixels by 598 is the patch size, and it's 20x. Yeah.
So I guess the challenge is you don't necessarily know what morphological characteristics you're trying to capture are for certain, when you're trying to predict certain gene expressions.
Exactly. I think it has to be done in a data-driven way, basically, to either evaluate a few different resolution levels for each gene then, or alternative, having this kind of multi-scale model to try to circumvent and capture all relevant information in one framework.
Great. So then the follow-on question from Dang is, what is the spatial resolution of the transcriptomic?
Yeah, exactly. So the predictions are defined by the patch size. And of course, you could imagine that you have different stride, so you kind of create a higher resolution. But in our case, I think we had maybe 50% stride, and we averaged over the overlaps and so on. But to be honest, I think that there's probably a limit to how precise predictions you can get. That it seems like when you average over the slide level predictions are like averaging over all patches. So that's one level of aggregation. So there's, I think on the local pixel level, there will be so much noise. So the moment I think that, yeah, the patch size, or maybe even combining a few patches, is probably the limit as we see it, but I don't know.
You also talked about tumor heterogeneity. So I wonder how much heterogeneity do you think there is in the expression associated with a particular slide? Because if you're just trying to predict one value, do you think that's effectively capturing this heterogeneity?
No, exactly. No, I think there's a lot of variability actually, and that's been shown by Spatial. So there are the spatial transcriptomics platforms that do the multiple ones of them out there. The one we used have this like relatively low capacity in capturing spatial resolutions, and like a grid, it's like hand-picked regions of interest. But there are also like other techniques from 10x Genomics and some like homebrew platforms as well that capture like a more spots where each spot can be get a degree of actual RNA-seq quantification. And those studies have revealed a lot of spatial variability within tumors. But there are, of course, some patients have tumors that are relatively homogeneous, and others have more heterogeneity, as we also know that there are like some patients with more subclones on a molecular level, and other patients that have a more molecularly homogeneous profile as well. So I think this is actual variability on the molecular level that is present. But the question is, of course, how much of that can we capture through this kind of indirect way, like predictions from H&E slides? So some of it, but maybe not all.
Yeah. So Darwood has asked, the expression profile of a group of genes might be correlated. Have you tried using a single model for predicting the expression of all genes at the same time?
Yes, we did that. We have done that, and we do. So you don't get as good results. I can't remember. There's an archive paper on by Weitz et al., which is from us as well, using just TCGA prostate cancer data set, where we compare different strategies to get more computationally efficient prediction of gene expression values. And one of the models are this kind of multivariable response where we have all the gene expression values as the response, and we predict them in a single model. And that performs clearly worse than alternatives, like if you have single gene models, or in that paper that is, I think, accepted now for publication in bioinformatics, that show that you can cluster the genes first and group them into some subsets based on co-expression patterns, like on the RNA-seq level, and then predict such clusters that seem to be working quite well. And then you have reduced the computational cost substantially compared to the like quite brute force approach that we took in this first paper.
Great. Is there any questions in the room? So we're joined as well. There's around 12 people in the room as well. Who's joining. So there's no questions in the room. I don't think any more online. So I think with that, we can thank you for taking the time to give this excellent presentation. Some brilliant work that you and your team are doing, and have a great rest of your day.
Great. Thank you so much for having me, and thanks for listening in. So yeah, thank you.
See you.